Hello and welcome. In today's deliberation, we shall talk about the construction of genomic and cDNA library, and we shall also try to understand the importance of genomic and cDNA library. First of all, let me give you a brief introduction about DNA library. Complete knowledge about the whole genome of an organism led to many developments and innovations in the field of molecular biology. But there was always a difficulty in studying the DNA of interest because of its availability, and then molecular biologists found a solution to this problem by constructing gene library, which holds the collection of DNA sequences derived from various organisms. A DNA library is a collection of DNA fragments ligated into a cloning vector and stored individually in a host cell. Libraries are important resources for molecular cloning and genome sequencing projects. They can be formed from genomic DNA or messenger RNA cDNA, that is complementary DNA. Insert size can vary over several orders of magnitude, hundreds of base pairs or more, and they can be maintained in yeast or more commonly E. coli. Genomic library contains the DNA extracted from the nucleus of a selected organism, while in case of a cDNA library, the mRNA of an organism is turned into cDNA, complementary DNA, with the help of reverse transcriptase enzyme. Both the genomic and cDNA are then subjected to fragmentation, then ligated to vector, and then introduced into the host bacterium. The group of transformed bacterial cells holds the DNA of selected organism and is stored. A genomic library holds the entire DNA sequences of an organism, whereas the cDNA library holds the expression gene sequence of the organism, as it is generated from the mRNA of the organism. Now, let's talk about the genomic library construction. Cloning an entire genome in the form of a library of random genomic clones without identifying them is often called as shotgun experiment. In this experiment, genomic DNA is extracted, broken into fragments of a reasonable size by restriction into nucleases, and then inserted into a cloning vector using DNA ligase to generate a population of chimeric vector molecules. A set of fragments cloned in this way is called a genomic library. Once such library is available, then clones can be propagated indefinitely in a plasmid vector and a gene when a specific probe is available. Here are the steps for creating a genomic library from a large genome. First, extract and purify the DNA. Second, digest the DNA with a restriction enzyme. This creates fragments that are similar in size, each containing one or more genes. Third, insert the fragments of DNA into vectors that were cut with the same restriction enzymes. Use the enzyme DNA ligase to seal the DNA fragments into the vector. This creates a large pool of recombinant molecules. Fourth, these recombinant molecules are taken up by a host bacterium by transformation, creating a DNA library. Now we will learn how to determine the titer of library. After a genomic library is constructed with a viral vector such as lambda phage, the titer of library can be determined. Calculating the titer allows researchers to approximate how many infectious viral particles were successfully created in the library. To do this, dilutions of the library are used to transfect cultures of E. coli of known concentrations. The cultures are then plated on agar plates and incubated overnight. The numbers of viral plaques are counted and can be used to calculate the total number of infectious viral particles in the library. Most viral vectors also carry a marker that allows clones containing an insert to be distinguished from those that do not have an insert. This allows the researchers to also determine the percentage of infectious viral particles actually carrying a fragment of the library. A similar method can be used to titre genomic libraries made with non-viral vectors such as plasmid and BACs. BACs are bacterial artificial chromosomes. Genomic libraries can be prepared by using a number of restriction endonucleases one at a time, so that fragments of varying sizes having cuts at different places of the genome will be available. However, this may lead to cuts at inconvenient places, including sites within a gene, so that fragments having complete gene will be difficult to obtain. In order to overcome this difficulty, we use the following strategy in the shotgun experiment. First, restriction endonucleases are used, which have long, maximum 8 base pairs recognition sequences, so that such a sequence may be infrequently distributed. Second, conditions are used which give only partial digest, so that a particular restriction site is only occasionally cleaved, and long fragments without having any breaks on recognition sites are available within a gene can be easily obtained. Now we will shift towards the cDNA library construction. cDNA, that's complementary DNA, is produced from fully transcribed mRNA found in the nucleus using reverse transcriptase enzyme. cDNA library is a combination of cloned cDNA fragments inserted into a collection of host cells, which together constitute some portion of the transcriptome of an organism. The advantage of cDNA library is that it contains only the coding region of a genome, as the mature mRNA in eukaryotic cells is already spliced. Now, to prepare a cDNA library, the first step is to isolate the total mRNA from the cell type of interest. Because eukaryotic mRNA consists of a poly A tail, they can easily be separated. Then the enzyme reverse transcriptase is used to synthesize a DNA strand complementary to each mRNA molecule. After this, single-stranded DNA molecules are converted into double-stranded DNA molecules by DNA polymerase. They are inserted into vectors and cloned. Now, the choice of vector, what kind of vector we should use. Cloning vectors are extra chromosomal DNA which can replicate in the host. Vectors have physical properties that facilitate purification separately from the chromosomal DNA of the host. Vector choice for library construction will vary by the use and the goal of the library. The most important criteria is the desired size of the insert. A good vector must have following properties. First, it should be able to replicate autonomously. Second, it should be ideally less than 10 kb, that is kilobase pair, in size, as larger DNA present difficulties during gene cloning. Third, it should be easy to isolate and purify. Fourth, transformation of host with vector should be easy. Fifth, it should have the ability to integrate itself or the desired DNA fragment to the genomic DNA of the host cell. Last, a vector should have as many as possible restriction sites for restriction endonucleases. Now, the various types of vectors. A genome size varies among different organisms, and the cloning vector must be selected accordingly. For a large genome, a vector with a large capacity should be chosen so that a relatively small number of clones are sufficient for coverage of the entire genome. However, this is often more difficult to characterize and insert contained in a higher capacity vector. Here are some of the vectors commonly used in constructing libraries. First, plasmids. A plasmid is a double-stranded circular DNA molecule commonly used for molecular cloning. Plasmids are generally 2 to 4 kbs in length and are capable of carrying inserts up to 15 kbs. Plasmids contain an origin of replication, allowing them to replicate inside a bacterium independently of the host chromosomes. The plasmids commonly carry a gene for antibiotic resistance that allows for the selection of bacterial cells containing the plasmids. Next type of vector is phage lambda. It is a double-stranded DNA virus that infects E. coli. The lambda chromosome is 48.5 kb long and carry inserts up to 25 kbs. These inserts replace non-essential viral sequences in the lambda chromosome, while the genes required for the formation of viral particles and infection remain intact. The insert DNA is replicated with the viral DNA. Thus, together they are packed into viral particles. These particles are very efficient at infection and multiplication, leading to the higher production of recombinant lambda chromosomes. Third type of vector are cosmids. These vectors are plasmids that contain a small region of bacteriophage lambda DNA called the cos sequences. This sequence allows the cosmid to be packed into bacteriophage lambda particles. Cosmids are capable of carrying inserts up to 45 kbs in size. Now, how would we screen and select our recombinants? Library screening relies on information available about vector and genes of interest. In this, clones of the library are subjected to analysis to identify desired sequences. Different methods are used to identify clones harboring fragments of genes of interest in the library. DNA sequences can be screened for the gene sequence of interest among the clones of library with oligonucleotide probes that match to the sequence of the clone of interest. Functional screening for the encoded protein products by the expression of the gene of interest can also be performed to identify it. Now, immunochemical methods of screening. The desired gene can also be identified by the activities of the protein products of the encoded gene. This type of strategy is used for cDNA expression libraries. The fragments are cloned into special cloning vectors which allow the functional expression of cloned DNA fragments. The protocol of immunological screening is similar to hybridization screening, except the fact that antibodies are used as probe instead of nucleic acid. Clones of the library are spread on replica plates. Transformed bacterial colonies on Petri plates are lysed to release protein antigen from the positive colonies. Protein samples are transferred to similar position on the nitrocellulose membrane so that the pattern of the clones remains same on the membrane and the replica plates. Released bacterial and expressed proteins are bound to the membrane. Membrane is overlaid with primary antibody which binds to the protein of interest. A secondary antibody with a detection system is then overlaid on the membrane to detect protein primary antibody complex. Unbound antibodies are then washed out. Bound antibodies are detected as it produces a colored compound. The colonies with positive results are isolated to identify the vector and correct gene of interest. The limitation of this technique is that it can be used only when specific antibody is available. Now, another type of screening is nucleic acid hybridization. Once a genomic or cDNA library is available, we may like it to use for isolation of gene sequences. This can be achieved by nucleic acid hybridization technique, where denatured DNA fixed on nitrocellulose filters is hybridized with radioactive probes. Nucleic acid hybridization can be carried out by two ways. First, colony hybridization. In this technique, bacteria carrying chimeric vectors are grown into colonies which are lysed on nitrocellulose filters. DNA is denatured in situ and fixed on the filter, which is hybridized with a radioactively labeled probe carrying a sequence related to the gene to be isolated. Colonies carrying this sequence will be identified by the dark spots after autoradiography, so that the original chimeric vector carrying the desired gene sequence can be recovered from one or more colonies in the original master plate and used for further experiments. This technique is described as colony hybridization. It is possible that a probe may identify more than one clones or that a gene is fragmented in a library. In such a case, one needs to reconstruct the desired sequence using several overlapping sequences available in the library. This is a very routine exercise whenever we like to isolate the specific DNA sequences from a genome of a species or from the cDNA derived from mRNA of a specific tissue of species. Now, the plaque hybridization. Sometimes a library may be available not in the form of bacteria transformed with chimeric DNA molecules, but in the form of phage particles carrying the cloned segments. In such a situation, a bacterial lawn is infected with a mixture of chimeric phage particles, that is the library, and a large number of plaques develop overnight. These plaques can be treated just like the colonies in the colony hybridization to identify and isolate the chimeric phage particles carrying a gene of interest. Now we will shift our focus towards the gene probes. A gene probe is a technique developed in 1980s that involves a fragment of DNA or RNA which has been labeled with a radioactive isotope or a fluorescent marker. It is used to detect the presence of a particular sequence of bases in another piece of DNA or RNA. These probes are useful in the identification of abnormal genes responsible for hereditary diseases such as cystic fibrosis or Huntington's disease. Identification of abnormal genes is achieved by mixing the gene probe with a piece of the DNA being investigated. Because the probe is a single strand, it seeks out its complementary base sequence and binds to it. In this way, the probe locates exactly where the target gene is. If the gene is normal, it will bind with the probe and produces a normal sequence. If the gene is abnormal, the probe will not bind and a gap in the DNA will result. This shows up on the gel when the DNA is sequenced using electrophoresis. Now the southwestern screening. Southwestern screening, also known as southwestern blot mapping, is performed for rapid characterization of both DNA binding proteins and their specific sites on the genomic DNA. Proteins are separated on polyacrylamide gel electrophoresis, also known as PAGE, and blotted onto nitrocellulose membrane by diffusion. The genomic DNA region of interest is digested by restriction enzymes selected to produce fragments of appropriate but different sizes, which are subsequently end-labeled and allowed to bind to the separated proteins. The specifically bound DNA is eluted from each individual protein DNA complex and analyzed by polyacrylamide gel electrophoresis. Evidence that tissue-specific DNA binding proteins may be detected by this technique has been presented. Moreover, their sequence-specific binding allows the purification of the corresponding selectively bound DNA fragments and may improve protein-mediated cloning of the DNA regulatory sequences. With that, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.